This is episode 195 of the STEMCELL podcast, the Global Alliance for iPSC Therapies with Dr. Stephen Sullivan. Hey everybody, we are Dr. Daylon James and Dr. Arun Sharma. Welcome back to the STEMCELL podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. As you know, the ISSCR 2021 annual meeting is taking place from June 21st to the 26th. And as always, we have big plans to bring you the latest research being presented at the meeting. Throughout the week, we will be releasing daily episodes discussing some of the meeting's highlights and chatting with special guests, including Doctors Chuck Murray, our friend, our other friend, Dr. Madeline Lancaster, and other friends. What's more, we will be releasing these episodes not only in their traditional audio format, but also as video episodes, which you can find on the STEMCELL podcast website or STEMCELL Technologies YouTube channel. So stay tuned for that. I'm going to have to get a haircut and shave. Today, we have Dr. Stephen Sullivan from the Global Alliance for iPSC Therapies. He's on the podcast to talk about how the initiative supports the implementation and clinical application of therapies derived from pluripotent stem cells. We've also got our usual roundup of recent highlights and stem cell news coming right up.
But first, we'd like to remind our listeners about ESC and iPSC News, which, if you haven't heard by now, is a free weekly newsletter brought to you by STEMCELL Science News, summarizing all of the latest research, news, jobs, and events in the ESC and iPSC fields. So save time and keep current with ESC and iPSC News. You can subscribe for free at www.ixcellnews.com. We're going to talk about a cell stem cell paper for the first paper of the Roundup here, and it's actually coming from a PI who we've covered previously and is one of our favorite names to say in science, Dr. Panteleimon Rompoulas from the Perelman School of Medicine at UPenn. First author is Xixia Huang. Title of this paper is LGR6 marks epidermal stem cells with a nerve-dependent role in wound reepithelialization. We're talking about LGR6, not LGR5, which is that famous marker of intestinal stem cells, if I understand that correctly. So we know that stem cells, of course, support the maintenance of a bunch of different organs, skin in addition and included. But we got to talk about injury and how injury is inducing stem cell differentiation, stem cell function to actually restore lost cells. And again, we're talking about the skin, and these folks have shown that LGR6 is marking a regionally restricted population of the epidermal stem cells that are actually, the neat part of this is these stem cells are interacting with nerves. And it's this interplay between the nerves and the skin stem cells that actually is contributing to wound healing and reepithelialization. What they did is they actually used some diphtheria toxin to ablate the LGR6 population and found that when they did that, it delayed wound healing, as you might expect. And the nifty thing is when they denervated the skin, removed the nerves, it also phenocopied that effect. So they used a lot of really neat intravital imaging as well to figure out how these stem cells actually move around and repair the skin after injury, and how losing the nerves can actually kind of delay that process. And that these LGR6 positive stem cells are really critical to the reepithelialization. And it's, again, this loss of the nerve populations in the skin is also going to phenocopy this loss of the LGR6 population. So the induction of the reepithelialization process is also inducing the recruitment of other stem cell populations. Apparently the skin has way more than just this one population of stem cells, including the hair follicle stem cells, which, if I remember correctly, we're actually going to be talking about in the next, in one of the Roundup papers here. We've actually got 2 papers related to skin and 2 papers related to the blood. And so when these hair follicle stem cells are being recruited, they are compensating to actually help mediate the wound closure. So they are working together with the LGR6 population to ultimately close the wound. And of course, you got to do some single cell if you're talking about wound healing, if you're talking about different populations of the stem cell populations in the skin. And they found that the fate of the LGR6 positive stem cells is shifted towards a differentiation profile after they lose their niche. All right. So the big picture here is that this LGR6 population is a population of epidermal stem cells that are needed for the injury response. They interact with other stem cell populations in the skin, and they also interact with the nerves to regulate the ultimate fate of skin renewal and skin regeneration. And I think, we've talked a lot about this recently on the show, especially in the context of the skin, which I think is a fantastic model system for studying this. The importance of the niche, all right? It's not just the stem cell intrinsic properties that are contributing to the regeneration of different organs like the skin, but it's also the contribution of other components in that niche, like the nerves in this particular example. So another neat study looking at skin regeneration. I thought we had figured out all the different stem cell populations in the skin, but hey, LGR6 is another one.
Yes, this, as you alluded to in the end there, the niche, it's complex, and it's there's this dichotomy. I don't know if it's a dichotomy, but there's a kind of two sides to the coin here in stem cell science, where we make these organoids, right? And we talk about how we're approximating, organ function, and then you see the actual niche in the cells in the animal, and you realize, or at least it underscores how far we have to go with organoid culture to really get there. But I mean, Pantaleimon, he's on his way. Last month, cell stem cell, this month, cell stem cell. He is a alumnus of the Greco lab at Yale, NICEF, New York Stem Cell Foundation, Druckenmiller Fellow, like myself, I am proud to say. So yes, Pantaleimon, he is on fire. And the name, I thought it was like Spanish. I mean, this is a stem cell show, but I'm going to tell you a little about etymology. I don't know why not. The name actually means merciful. It's a Slavic name. So, be more like your name, Pantaleimon. Have mercy on us. Why don't you leave some of the science for the rest?
He's doing a fantastic job. Their lab is on fire, and back-to-back stem cell stories. You mentioned the organoid angle, right? Organoids are, of course, talked about a lot on this show, and one particular class of organoids are the intestinal organoids, which are marked actually by the next door neighbor of LGR6, LGR5. And this is actually something that came up here in this particular paper. They're wondering about the leakiness of some of their reporter lines here. Would the LGR6, LGR5, are they close enough when it comes to the protein structure, the overall sequence? Are they close enough to actually cause some leakiness in the expression of some of these Cree mouse lines that they looked at? So, and fortunately, it didn't look like there was that leakiness, but it tells you also a little something about, genes that you might think are so closely related just by the basis of the fact, their sequence is very similar, LGR6 versus LGR5, but they can perform entirely different functions in the body. Another example of that in the context of the work that I do is the GATA family of transcription factors, GATA5, GATA4, GATA6. They are expressed in different places. Like GATA6, for example, is expressed in the endoderm and also the pancreas, whereas GATA4 is one of the master regulators of the heart development. So, that one little number, LGR6 versus LGR5, makes a big difference.
A world of difference. And you mentioned the tools, the Cree drivers and all the stop flocks and lineage tracing genetically engineered mice that we use. Which are, an issue. It's not an issue, but they have their limitations, as you kind of spoke to there. And the investigators here, Pantaleimon and his group, were very careful in showing the fidelity of these. But, there's a lot of tools that have been developed for lineage tracing. I'm staying in the epithelium here. We're going to talk about another story with epithelial stem cells and lineage tracing and imaging. And this, stems from that idea that these are tools and they don't necessarily tell you the truth. They just give you some indications in that direction. And it's talking about epithelium and ectodermal lineage and differentiation. Most of ectodermal appendages, including the hair follicles that we just talked about, we're going to talk about again, they come from the placode, okay? And a lot of previous studies have indicated that this gene SOX9 is expressed in cells that are generated by asymmetric division of epithelial cells in the placode, and that's what leads to becoming these bulge stem cells, okay, the hair follicle stem cells, right? But it's not really certain, it's not clear how an immature placode develops into the mature hair follicle structure, right? And one of the problems and the obstacles to really understanding that and seeing it is that you don't really know specific markers that enable this prospective identification of all the cell lineages amongst the placode and the derivatives, right? There's a lot of adult stem cell markers for hair follicle stem cells, including like SOX9, NFAT-C1, LHX2, keratin 15. But there's a lot of spatiotemporal variation in the expression of those markers, because these are like ancestral, like in the case of Cree and the stop blocks. If it turns on, it stays on. So it's hard to really tease apart the timing and the progression of these cell lineages. So Hironobu Fujiwara from the Riken in Japan and his group, they endeavored to address this by using just a brute force, long-term ex vivo culture imaging system of whisker hair follicles from mice. And this was the key here, is a technical innovation where they were able to image these things starting at embryonic day 13 plus 60 hours. So they had long-term extended imaging of the hair follicle to the bulbous peg stage. Okay, so that's E13 plus about 2.5 days. All right. So, and we talked about this in the last episode too, and we've been talking about it. The imaging is really progressing to the stage where we can see things real time and over extended intervals. And combined with other technologies like single cell transcriptomics, we're able to really add new depth to analyses that have already been done. I mean, we've been looking at these systems for a long time, but now we're adding increasing temporal, spatial resolution as well as transcriptomic. And that's what they did here. They combined that long-term imaging with single cell to show the progression of the entire epithelial cohort within the mouse hair follicle during development in real time, and showed that, and this was really cool, they showed that the precursors of the different epithelial lineages, they were aligned in kind of a concentric manner in the hair placode, and each ring had its own identity, and they acquired a unique transcriptional character and then extended into these cylindrical compartments that were aligned. And the bulge stem cells were derived from the peripheral ring, which is contrary to what everyone thought they knew. They all thought it was from the suprabasal cell. But here, Fujiwara and group showed that it was actually from the peripheral ring. They also showed that there was the 13 gene clusters that drew the entire transcriptional landscape of epithelial lineage diversification, at least in embryonic stages, writ large. All right. So then they went really off the wall and took historic work from insects, okay? And they converged on this generalized telescope model they call, where it's essentially these concentric rings in the placode that telescope out to form these aligned 3D longitudinally aligned cylindrical compartments. So it's not just like a story about imaging and looking at what happens in the epithelium of the hair follicle and the whiskers here, but it's kind of like talking about general, across, we're talking about insects. So, I mean, you'd have to go to yeast to get much farther, but this is like fundamentally a fundamentally conserved element of animal embryogenesis, it seems, in the ectodermal lineage. So it's a big deal, not just in terms of stem cell organogenesis, but also evolutionary comparative biology.
Yeah, I do want to bring it back to the tech development for a second. I mean, you talked about it. Imaging has taken this field and every field in biomedical research by storm. I mean, we talked about two photon recently, all these amazing developments in 3D live imaging as is described here, and of course coupled with the single cell, really gives you a resolution in development that hasn't been possible before. And I was thinking about it. We've been studying development for a very, very long time. I mean, the initial Xenopus experiments and Xenopus observations were made in the early 1900s, late 1800s even. But now that we have all of these amazing imaging technologies, part of me thinks that we have to revisit almost everything with not necessarily with human development, but just looking at development in general. I just feel like there's so many things that we've missed when it comes to the localization of different stem cell populations, cell migration, cell differentiation, so much to look at with all these new technologies. I mean, I get it. It's we don't want to reinvent the wheel, and of course the observations that we've made in these seminal studies over the years have stuck around for the most part. But I feel like we have to take another deeper dive into some of these developmental approaches and developmental processes, don't you think?
Absolutely. I think I don't know if it's a old saying, but it ought to be. All knowledge in science is provisional and needs to be reconsidered pretty regularly. And technology gives you a great lever there to look at questions that we know are important, but to add higher resolution. So talk about standing on the shoulders of giants. We can really revisit old questions and shed new light on them. So it's exciting to be in science at this time with all the tech around, isn't it?
It is, and you set me up because we're talking about tech in the next story as well. The next story is coming from the lab of a genome editing mastermind. This is focused on base editing. This is the lab of Dr. David Liu over at the Broad Institute. First author here is Gregory Newby. The title of this paper is Base Editing of Hematopoietic Stem Cells Rescues Sickle Cell Disease in Mice. And I believe Dr. Liu is going to be one of the main speakers and one of the keynote speakers at the ISSCR annual meeting, which of course we'll be attending in a few days. Here we're talking about yet another application of base editing. This is a technology that's really been pushed forward by the folks at the Broad Institute and Dr. Liu in particular, and I think they have a few startup companies that have actually spun off of the work that's being done in the Liu lab. So sickle cell, we're talking about sickle cell here, right? So they're basically applying their custom adenine base editors that they term ABEs towards different diseases that have point mutations that are caused by point mutations. And in this situation, they're looking at sickle cell, which is caused by a specific point mutation in the beta globin gene HBB. And they used a base editor, or ABE, to actually convert the sickle cell disease allele, which is dubbed HBBS, into a non-pathogenic variant, HBBG, also known as the Makassar beta globin. All right, this is reflecting actually one of the limitations of these base editors. It's not like you can convert every single base, one of the four bases into one of the other four bases. There are some limitations in terms of the patterning that you can do and in terms of the coding that you can actually change. So in this situation, they weren't actually able to convert the SCD or sickle cell disease allele into the wild type. They actually had to convert it into a non-pathogenic variant. So just a little something to note there. What they did is they actually delivered the mRNA encoding the base editors with a targeting guide RNA into hematopoietic stem cells from patients with sickle cell disease, and they saw an 80% conversion of the allele gene into the non-pathogenic variant. In 16 weeks after actually transplanting these edited human HPSCs into the immunodeficient mice, the frequency of this disease allele was, of course, very low. The frequency of the edited allele was around 68%. And the important thing was the function. They significantly reduced the hypoxia-induced sickling of the bone marrow reticulocytes fivefold. And that's really important because that means that this gene editing seems like it's a permanent fix. They looked at the physiological effects of the base editing, and they saw that there wasn't anything off target, which is part of the reason why base editing is being adopted these days. It's a much cleaner system than the traditional CRISPR-Cas9, which induces a double-stranded break, which I like to dub taking a shotgun to the genome and just kind of blasting it. It's not the cleanest process, okay? So after 16 weeks, this Makassar beta globin, which is the non-pathogenic variant, actually represented around 80% of the beta globin protein in the blood of these mice. And they, the hypoxia was still reduced significantly. They showed very normal like blood parameters, reduced pathology in the spleen compared to the mice that received the unedited cells. And then they did a secondary transplantation of the bone marrow, confirming again that the gene editing was durable in long-term transplanted hematopoietic stem cells. So all in all, I think this is a tech-centric paper showing the importance of base editing as a really clean editing strategy for specific pathogenic variants. It's not applicable to everything because, as I mentioned, this isn't a way to edit every single base into every single base. It's only specific situations that this is applicable. But when it is applicable, it is a clean fix, no off-target edits. And I think this is really taking the genome editing field by storm.
This is huge. I mean, a lot of the stories, it's been one huge story after the next. We just covered a couple weeks ago, you talked about the ixKID, or one of those types of, immune deficient diseases that was addressed by genetic engineering. I mean, this is the thing decades ago we talked about. We were waiting for it to come, I think, a little bit sooner, but now it's just everywhere. And I think that this, we're curing people effectively. We societally are curing people of sickle cell already right now using the technology that has been hailed for decades. And now this is like 2.0, 3.0, whatever 0.0. So, and the other point that you make there, while it's not for every disease, there are specific diseases this is suited to. I think that the way they went about it here really is a testament to the ingenuity of scientists. It may not be for every disease, but you're not correcting, you're not restoring the native allele here. You're going a different way. You're bypassing. It's kind of like there was another hack I remember where you activate fetal globins to get around, a deficiency in the adult globin. So there's these hacks that I think, native isn't necessarily a cure all the time. You can find another way. And I mean, this is just so exciting, I think, to see that the therapies are finally in people, not this one, but it won't be long, and still improving. And, all the sequelae that you would worry about, like oncogenesis or cancer, in this case, have been addressed. So it's very exciting.
Yeah, you mentioned the ingenuity of the folks who are doing this kind of work, really clever approaches to bypassing the pathogenic variant and engineering a non-pathogenic variant. This kind of reminds me of what Eric Olson's lab has been doing in their studies of Duchenne's muscular dystrophy, where they actually did an exon skip strategy as opposed to a complete genetic correction of the gene that actually causes Duchenne's. But they basically decided to skip the causative exons that are causing the disease, sort of in a similar fashion to what they're doing here. Not a perfect fix, but it's going to do the job. I think base editing is something that's being readily adopted by different labs around the world, and part of it has to do with Dr. Liu's research strategy, which is make these reagents pretty readily and quickly available to anybody who wants them. They're depositing their plasmids onto Addgene very quickly, within oftentimes even before the paper is published. So I think that's a big reflection on to the openness of science, and in particular the openness of this particular lab in making science accessible to everybody around the world.
Scientists helping scientists, people. That's what they do in the best case. So yeah, we're talking about genetic engineering and therapy, and maybe CRISPR, in this case, base editing. And this has been proposed, since the Berlin patient with HIV. This is my segue here into another blood story. Since the Berlin patient, people have been talking about a long-term cure, not just suppression of the virus for HIV, right? By making, and this is the John K. Hay thing, which is a disaster, but same idea, is by making the immune cells kind of invisible to the HIV. And that's an idea. But just speaking about HIV right now, the therapies work, right? The modern antiretroviral therapies are enabling people with HIV to live longer. Life expectancies that are approaching those of people without HIV, right? So, effectively, you could argue that we've got a cure, or we've suppressed it to the point where it's no longer a scourge that it was, at least in the developed world. But HIV is also associated, and not many people know this, but increased prevalence of other comorbidities such as cardiovascular disease and malignancies that aren't directly related to the HIV or AIDS. Even with people who have been totally virologically suppressed, have been on antiretrovirals for years since first diagnosis, they still have increased comorbidities. And part of this may be related to clonal hematopoiesis. Okay, so clonal hematopoiesis, it's the overproduction of blood cells from a single hematopoietic stem cell that has a somatic mutation, and that gives that hematopoietic stem cell and its derivatives a clonal advantage. So you get these clones that kind of are more represented in your hematopoietic cohort. And it increases with age, as you might expect, because you get more mutations. And it's also associated with development of hematologic malignancies, also cardiovascular disease and stroke, and just general. All-cause mortality, clonal hematopoiesis is linked. So given the increased incidence of cardiovascular disease and malignancies in HIV, and then also separately the link between clonal hematopoiesis and malignancy and cardiovascular disease, obviously the connection is, hey, maybe HIV itself via clonal hematopoiesis is leading to these comorbidities. And to address this, it's a big group here. And this paper was led by Kathy Petumenos, Sarah Jane Dawson, Mark N. Polizotto, and Mark Dawson. They're from the Kirby Institute and the Peter MacCallum Cancer Center in Australia. And they made up this group called the Age-Related Clonal Hematopoiesis in an HIV Evaluation Cohort. Okay, it's called the ARCHIVE study. And they put together this study as a pretty massive, considering the care they took, in which they took 220 HIV positive and 226 HIV negative participants, all aged 55 years or older. They were recruited and prospectively monitored. Peripheral blood was collected serially collected to assess whether or not they had whether there was present any clonal hematopoietic mutations. And what they found was that there was a total of 135 clonal hematopoietic mutations identified in a total of 100 of the 446 participants. So roughly a quarter of all the participants had incidents of clonal hematopoiesis. And as you might expect, the clonal hematopoiesis was more prevalent in the HIV positive participants, with an odds ratio of about 2. The patients with clonal hematopoiesis were pretty much twice as likely to come from the HIV positive pool. And also both hematopoiesis, clonal hematopoiesis, and the HIV infection were associated with increase in biomarkers associated with inflammation. So 2 things there. It suggests that there's increased emergence of clonal hematopoiesis in HIV-infected individuals, and this may be related to a state of chronic inflammation related to the HIV infection. So, I mean, as far as stem cell stories go, this is much more clinical, but I think it's a really important milestone for the field to definitively attribute these comorbidities to HIV itself and possibly the inflammation. I think it's a really good lead there, the inflammation at least, as to what maybe the pathophysiology physiology underlying these effects are. So it would be fodder for a lot of future study and a nice clinical endpoint that definitively makes this link once and for all.
Yeah, I'm just learning about clonal hematopoiesis. I'm not really a blood guy, and unlike you, who is living and breathing blood, right? But when it comes to clonal hematopoiesis, it's hard for me to say it actually. Clonal hematopoiesis. When it comes to CH, I'm wondering if it's a causative relationship or is it more of an association? I mean, some of these mutations may not necessarily be directly contributing to the progression and to the prevalence of the disease. So I guess what I'm getting at, and something we discussed before the show, is this sort of a byproduct, or is this actually, really directly implicated in the causation of the disease?
Yeah, I think that, given this, I wouldn't say isolated study, there's some other evidence pointing in this direction, but I would say more it's the totality of the evidence over almost a decade, at least 5 years, that has suggested that there is in patients with clonal hematopoiesis a really broad constellation of cardiovascular and other sequelae, and in particular that is perhaps related to a myeloid bias amongst the hematopoietic clones. And so I wouldn't say in this study specifically, but I would say given other more mechanistic, and I would say studies with more molecular resolution showing the way that the hematopoietic clonal hematopoiesis can distort the pool, I think that maybe provides more of a basis or a rationale for the link. But yes, it's the age-old question anytime you do anything in science: correlation or causation. With the clinical study, it's really hard to say, but that's why we get into the mice next. Arun, we'll see. It won't be long. This archive study, I'm sure, is going to be, the root of a long scientific branch. And with that, the interview. This week, we're talking to Stephen Sullivan, and talking to somebody who's geared towards getting iPS cells out to the world in a sensible way. It's a good time to bring a message from Stem Cell Technologies. As research using pluripotent stem cells advances toward the clinic, there's a renewed focus on cell quality. Visit www.stemcell.com/cell quality to explore ways to assess your human pluripotent stem cells and learn about essential quality attributes to consider for gene editing, disease modeling, and maintenance. Okay, we'll talk to Stephen Sullivan about all those points in the interview coming right up. All right, guys. On this episode, we are delighted to welcome Dr. Stephen Sullivan, who's the Program Manager and International Liaison Officer for the Global Alliance on iPSC Therapies, otherwise known as GATE. GATE is an international resource to facilitate the therapeutic use of immunogenetically matched, clinical-grade new types of stem cells, induced pluripotent stem cells that we know about a lot, for the benefit of patients globally. GATE's vision is that patients globally will have equity of access to this new generation of iPSC-derived cell therapy products. Dr. Sullivan, thank you so much for joining us on this episode.
Thank you very much.
Yeah, well, thanks for being here, Dr. Sullivan. As Dalen mentioned, you're part of GATE, this global alliance for iPSC therapies, and it's no secret that iPSCs have tremendous potential for cell therapy. You can, of course, make a variety of different somatic cell types from iPSC colonies and differentiation protocols have improved tremendously over the last decades, as of course you're aware of. And we've discussed this plenty on the show. I mean, there's tremendous potential for these cells, but there are certain limitations when it comes to cost, applicability, making sure the lines we're using are universally accepted, GMP manufacturing, so on. There's a lot of issues to tackle. So where does GATE actually fit in here? And what specific steps are you taking to actually make sure that this universal and global mission of making iPSC therapies is, a realistic reality? So what are you guys focused on?
I suppose basically what we are is an organization that is there to facilitate consensus around key elements of quality. So, if you look at other types of stem cells research and translation, sometimes there's not a pivotal coalescence around what constitutes quality or what constitutes a type of stem cell at the beginning of the sector's development. And this then drags into people thinking that they're on the same page, using the same definitions, but actually they're not. And this means then that they think they're working in a coordinated fashion, but then when it comes to actually manufacturing new therapeutics, they have huge problems. So GATE has spent a lot of time focused over the last 4 years on building consensus around critical quality attributes. So what is a clinical grade iPS cell, i.e. what is an induced pluripotent stem cell that is going to be used as a starting material for therapeutic development. And we had a series of workshops, and then we published the first iteration of those critical quality attributes. And the idea is over time we will refine those and gradually lift them up so that the sector as a whole becomes more and more aligned. So if you think about any type of cell therapy, some of the biggest risks to actually getting that to market are things like financial inviability or adaptation of different technologies at different time periods, and also regulatory discordance. And these are things that are generally not thought of to any great degree by early translators in on the way to the clinic. And what GATE is seeking to do is build a more holistic vision of what is ultimately required to get new iPS cell therapeutics to the clinic. So that if you build a holistic vision for translation and people know what are the key bottlenecks to getting stuff to the clinic and helping the patient, we'll all be better off, because the more therapeutics reach the marketplace and the more patients that are assisted and helped, then ultimately the more consistent funding will come into research over time. So everybody benefits if there is efficient translation to the clinic.
Yeah, GATE, it's funded by a global consortium of organizations, right? I mean, that's the whole point, is that you're getting everybody on the same page. That includes the New York Stem Cell Foundation, the Cell and Gene Therapy Catapult, Center for Commercialization of Regenerative Medicine. Those are in New York and London, Toronto, Canada. Also Korean HLA-typed iPSC Banking Initiative in Korea, Seoul. French Institute of Health and Medical Research, INSERM, that's in Paris, of course, and then the University of Hong Kong. I mean, there may be others, but that in itself right there, that's the world pinnacle institutions on all the greatest, I would say, academic centers, right?
That's just the board. Yeah. Our membership is even broader. It's more like 500 to 600 members.
Wow.
So the board, I think, is obviously very important, because many strategic decisions are taken at board level. And when you're talking about the likes of Michael May and Susan Solomon and the other directors and chair, there's a very deep well of knowledge pertaining to the stem cell sector. And one of the key things is having that memory of how things didn't go efficiently in the past with other types of stem cells or other cell therapies. And that's really quite crucial, because if we make the same mistakes repeatedly, with each cell type or with each new therapeutic, then there's a huge amount of waste and confusion. And it's important that we remember how things went wrong in the past so that we make new mistakes going forward. Right. And there's always a lot of confusion if things like critical quality attributes for iPS cells are not worked out early on before the sector really starts to explode. So, I think that, we're waiting on, like, obviously the iPSC sector is growing quite fast already. And that's, because of, immune oncology's, explosion. And, there's a lot of interest coming from the MSC space. So, Sonata using iPSCs instead of primary material to make their MSCs. So there's a lot of kind of what I call indirect forms of funding coming into the space. But when we get the results of clinical trials, if in human efficacy data is published, then that's really going to be when the IPS sector really takes off even stronger than it has already.
Yeah, you talk about quality a lot. It's come up a couple times already in this conversation. And of course, a big part of making iPSC-based cell therapy a reality is quality control to make sure the cells you're using for therapy meet appropriate, criteria and regulations for, say, GMP grade manufacturing. And one thing you're actually doing right now is collaborating with STEMCELL Technologies to actually publish a study on quality control for iPSCs. So we'll actually call you back on the show down the road once that's published. But why don't you give us a little bit more insight into that particular study, this collaboration? What do you think the most critical criteria are for quality assessment for iPSCs, and what are you learning?
Well, if I'm think about it. If I'm sticking cells, if I'm injecting cells into you that are derived from an iPS cell, you want to be very certain that those cells reach particular specifications. So things that they're sterile, for example, or that the cells are not genetically unstable and then start behaving aberrantly and causing cancers and such. We were initially, like 4 years ago, we had this quality workshop where we asked our membership how they defined and tested for iPS cell quality. And we took the answers and, basically we canvassed 18 different institutions, and we got 18 completely different answers. So different assays were being used. If they were using antibodies, different antibodies were being used, and even different definitions about what specifications were required to give you a positive result or a negative result when doing a quality test. So this really showed to us that, people thought they were considerably more aligned about what constitutes quality and testing for quality. They thought they were way more aligned than they actually were. So you could almost see, if the field didn't acknowledge this and try and coalesce, and test the assumptions around, okay, are our quality tests consistent and indicative? The field would end up having big problems in that I won't be able to, if I take cells from your lab and I'm trying to make a therapeutic from it, I will, have a very frustrating time because your tests are not the same in real terms as mine. And so what we did was, over the last 2.5 to 3 years, we collaborated with Syra in Japan and NIPSI in England and STEMCELL Technologies in Canada, as you say. And we held an induced pluripotent stem cell quality assessment round. So this consisted of us shipping blinded standard materials to 18 sites in 11 different countries and asking those 18 participants to test the standards we had given them. They didn't know what they were. And then to give us back their data and their conclusions. And then we would amalgamate that data together. So individual participants in the quality round could see whether their quality data was consistent with what other people had done on these other sites, or whether it was an outlier. So basically, it tells you an awful lot about how consistent and indicative your quality testing is relative to the other sites. So we had, we had a really strong response when we had an open call for participation. We thought we would have 5 or 6 institutions, but we ended up having 18, which vary from like the Bioprocessing Technology Institute in A-Star in Singapore. I suppose large biotechnology companies like Blue Rock Therapeutics, Cell and Gene Therapy in the UK, obviously CSIRA in Japan, and CCRM in Canada. But we also had other centers like the Center for Regenerative Medicine in Barcelona and the Center for Stem Cell Research in the Christian Medical College in Vellore in India. So there was this really big mix of different types of companies like Fate Therapeutics, all the way down to universities that were very focused on clinical translation. So, we had 2 Brazilian universities who were involved, the Federal University of Rio de Janeiro and São Paulo University. And what was really good as well is we also had other leaders in the field like Fujifilm, CDI, and as INSERM in France, and other participants in Australia, so the Murdoch Children's Research Institute there. So there was a very broad mixture of participants that went through this process, and we're really excited to put that data out in the public domain. Obviously, one of the things that was quite difficult was steering this quite complex endeavor through COVID. And, some of our participants were frontline, involved in the vaccination programs and dealing with the consequences of COVID. So, for example, the Scottish National Blood Transfusion Service, they participated and, managed to stay on board while, all the COVID disruption was taking place. So, we're very thankful to all the participants, including, companies like Yapkesi in France, for, staying on board and, allowing, us to get this over the line, because I think, no one saw COVID coming. And then when you're working across, 11 different countries, it really, it takes trust and it takes patience to get a complex endeavor like that over the line.
Right.
Yeah,
for sure. I mean, it's not a small scale thing. You guys are reaching out to all kinds of institutes, as you alluded to there, and specifically named. I was having a look at the website. It seems like some, at least, of the practical applications in the workshops you have there are geared towards navigating the regulatory and consent aspects of generating iPSCs. Looking at this really drove home for me the challenge of regulating the clinical implementation of an individualized cell product, right? And of course, across many different cell types for many different indications, it's a challenge, yeah? And the question I think here is that we're talking about coalescing and aligning on standards before we get, into implementation, so we're not caught behind the 8 ball there. But it seems like, to me, this is constantly evolving. Do you think that by the time we arrive with some clinical applications of iPSCs that we will have a pretty solid set of regulations, or are we always going to have to keep these as provisional, evolving with every new therapeutic application that emerges?
Well, obviously the technology keeps evolving, but you got to remember that sometimes older technologies will get you regulatory approval quicker. And so when you move to kind of commercialization and wanting to have a clinical impact as quickly as possible, sometimes older technologies will do the job better and quicker than some of the newer technologies, whose, safety is still being assessed and characterized. So it takes time for new technologies to become familiar to the regulator, and sometimes newer technologies mightn't be cost effective at the beginning. So there's all these other elements that come into play when developing a therapeutic that are as important as the quality of the science. If those boxes aren't also ticked, then you don't get to the a product on you don't get a new therapeutic that actually you can roll out to large numbers of patients. So you alluded to the GATE website, which is gate.global, and membership is individual membership is free. And so there's several workshops there. One is developing a relationship with registries and identifying appropriate donor material. And that's really important, actually, because if you're taking donor material and reprogramming it to iPS cells and then making your therapeutic, the regulator will want to know an awful lot of information, not only about how you made your iPS cell, but they'll also want to know about the medical history of the donor, what drugs they were on, if there's a large, deep well of requirements from the regulator when it comes to these kind of cell therapeutics. And one of the things that GATE has is this deep understanding about transplantation and transfusion requirements. So obviously communicable diseases would be a big concern. So you want to make sure that your donor hadn't been exposed to various pathogens, for example, or they didn't have a lifestyle issue that meant that they their health was impaired, for example. If they were on drugs. So say, for example, you were taking an iPS cell and making it into a dopaminergic neuron, but the donor material that you initially sourced, the donor had been on a drug that was neurotoxic. The regulator is going to want to say, well, what's the effect of that when you put it into the patient? Is that going to make the therapeutic more risky than it needs to be? And so there's a lot of things to consider other than what's immediately apparent. There's one of the sayings at GATE is begin with the end in mind. You have to think through the whole translational journey, and sometimes it's good to have a place where you can meet other people from other silos who are equally important in the translational journey to find out what are the traditional bottlenecks in their area. So, for example, if you're an early stage researcher in academia, you're rewarded for ideation, novelty, your publications, and then when you get more into a scalable manufacturing mindset, you're thinking about can you grow the cells in large number? What number of cells would you want to elicit a positive clinical effect? So, if I have to grow up swimming pool, volumes of stem cells to treat one patient, then obviously that's a lot less feasible than if I have to, have a handful of cells in order to elicit a positive clinical effect. So, all these things come together, and then there's other elements that, clinicians would be talking about usability. When I develop a therapy, can a general nurse administer it to a patient, or do I need a really highly qualified, specialist? Because if I need a really, highly skilled specialist to administer the therapeutic, then actually the clinical impact will be much less, because it's much harder to roll out in large numbers. So understanding elements that really affect the clinical impact of the therapeutic down the line are good to know, even if you're early stage of translation.
Yeah, I mean, there's a lot of moving parts, as you've alluded to, and I think one of the toughest part of this all of this, what GATE is doing is the fact that you have to deal with multiple institutions and multiple regulatory bodies across the world and spanning various jurisdictions around the world. Each country has its own regulations when it comes to iPSC and cell therapy approval, that sort of thing. So it can't be an easy task achieving a consensus across international boundaries, given the huge range of laws and regulations governing cell therapy. And I was thinking about the genome editing field because I feel like this is an interesting parallel, right? They have to go through a lot of these same hurdles and same regulations to actually even think about making genome editing based therapies a reality. So I'm curious to see what are the steps you've taken to actually make this kind of like worldwide consensus on iPSC quality a reality. It must be really tough, working with so many different institutions across the world with all these different regulations.
Well, I think if you have people who've experienced trying to make cell therapeutics before iPS cells, they will know how things can go awry and how ultimately your therapeutic development journey is almost destined to failure unless you coalesce with what others are doing. And if for nothing else, if you're coalesced with what your colleagues are doing, it'll keep costs down, for one. And that becomes hugely important when you move more into the engineering or the scalable manufacture of therapeutics. So I think veterans in the field know how not coalescing around standards and definitions, it just leads to confusion, it leads to waste, and also it makes life harder for everybody, because whether you're a researcher or a translator, society is looking to you to be part of the chain, if you will, that's going to get these therapeutics out the other end. So it's important to work in a coordinated fashion. It is a challenge, but ultimately what it comes down to is having a platform where everybody's welcome and you can talk things out. So it's not like GATE comes along and dictates, all right, these are the quality, the critical quality attributes, everybody adopt those. No, they're like open meetings and people are encouraged to come and discuss, and the discussion then leads to usually quite a broad definition. But even just having a broad definition of requirements is very, very valuable then, because everybody's on the same page already, and you can refine that then over time. I think one of the key things, if you wait, obviously it's very important to engage the regulator about, if you're going to develop a therapeutic, you have to engage the regulator, but the regulator is there to stop the patient being exposed to unnecessary risk. Their role isn't to align the field or keep costs down or make developing new therapeutics easier. It's up to the community itself to align itself and to learn from the past, if you will. So apart from the registries and the regulators, there's a lot of workshops that are also on the GATE website that are free. If you join as a member, all you need to do is give us an email address so we can see who's accessing the site, and then you can basically look at those workshops. And even from the point of view, like one of the projects we're working on with eBISC and Andreas Kurtz is about developing better searchable databases to find iPS cells for specific applications, for example. So whether it be research grade or clinical grade, and that's also really important, because people need to find the iPS cells that are fit for their purpose.
Yeah, I can see it really is the bottom line there, is that you guys are creating a space for everyone to come together, coalesce on an agreed and high set of standards. But at the same time, the commitment, you're balancing all these different cultural centers, right, as well as scientific academic centers, and the commitment to therapeutic application of iPSCs. is different in different places. I mean, I know it's a matter of national identity in Japan, where iPSC's hailed from with the Yamanaka, and they've set up a lot of funding apparatus and institutes to accelerate, I think, the translation. Did you find that the regional political or cultural influences can, lead to, I wouldn't say an unequal contribution of any one group, but do you find that the voices in the room maybe have different priorities, and you have to balance the enthusiasm of some with the conservatism of others and find a medium? Or do you just lead? You just say, this is the standard scientifically, and you guys got to get on board?
Well, I think there's a degree of you're enabling that consensus, and part of enabling that consensus is being aware of cultural difference. So you can't be, ethnocentric. Like if you try and do everything the Canadian way or the Irish way or the American way or whatever it might be, then you'll be excluding a lot of other different cultures. And a classic example, say you mentioned the Japanese culture, which is, I suppose, be quite distinct from the Canadian culture, for example, in that when you have a formal meeting, the Japanese will tend to make the big decisions after the formal meeting to save face, because that's, honor and having respect is very important in the Japanese culture. And if you don't pay heed to the cultural elements, then, it's like, it's then you won't get the full engagement of the international community, if you see what I mean. So you kind of have to be aware of what like sometimes you might have a lot of extroverts in the room who'll do a lot of talking, but actually the real gold might come from the introverts if you allow them to give you feedback then later on. So you need to have a system that accommodates everybody and how they prefer to interact and how to ask for things. I mean, obviously, patience and listening to others is the rate limiting step. But I think if you start off well, and when you build a network, it's about having one-to-one relationships and establishing trust that we're all on the same page. We all want iPS cells to have the biggest clinical impact possible. Like, I mean, that we're not just getting, no one's interested in getting to square 99 on the snakes and ladders board and then falling all the way back down again. We want to get to the end and get a therapeutic out the other side that is, safe and efficacious. And, this is obviously ambitious, but, I think we've just seen, vaccines being produced to COVID-19. And also, I think the COVID-19 narrative really shows us how interconnected we all are. So, I think it's in some ways, it's one of the silver linings from a very testing time that, if we collaborate, we can achieve a lot more. And I think ultimately, when you're thinking about any cell therapeutic, you'll probably be leveraging different materials and different infrastructures at different parts of the world. So, differences in individual countries can be leveraged to overall benefit of efficient translation. So you might find one country where the regulatory requirements are very systematic and very clear. So you might go to that country for regulatory approval and making sure that your donor material or your iPS cells or your intermediate materials are fit for purpose. But then you might find that it's easier to carry out clinical trials in another country that has an integrated healthcare system, which means that it's easier to enroll patients, for example. So, that differences don't just divide us; they actually make us better at certain things, and that can be leveraged. But it all comes down to not just focusing on the technology, but also focusing on what can be achieved through collaboration.
Yeah, definitely. And it's part of the reason that working in this iPSC field is so fun, is because it is an international field. And even myself as an academic, I've had the opportunities to travel around the world to listen to amazing speakers from different portions of the world doing incredible science. And like you said, we're all on the same page. Ultimately, we do all want to see this become a reality. iPSC cell therapies become a reality, and you've traveled that scope of science. You've started in the academic side of things, and now you're more on the business side of things. And we're actually always really interested in non-traditional career paths here on the Stem Cell podcast. Most of our guests are on the academic side of things, but I think we have a really unique guest here today. So we wanted to showcase what you've done throughout your career. You actually received your PhD in regenerative medicine at the University of Edinburgh, focused on nuclear reprogramming, and in the pre-iPSC days, actually. And then about a decade later, you decided to actually get your MBA and make this shift to GATE. So did you see this translational potential of iPSCs coming from a mile away, and you decided to actually get more involved in the business side of things because you saw that coming? So what was it that actually brought you to your current role, and how did you make that transition from academia to GATE today?
Okay, so that's three points would be, why did I get into pluripotent stem cells? And basically, I was in Dublin the year that Dolly was cloned, which showed that you could take a mammalian cell and make it revert back to a pluripotent state. And also Jamie Thompson derived human embryonic stem cells. And those two discoveries side by side, you could say, wow, that's going to be amazing, because, one of the key bottlenecks to, developing new therapeutics is getting access to enough material to either study the disease or, screen for new drugs or develop cells that can have a positive therapeutic effect. So I went to Edinburgh and worked in one of the labs associated with cloning of Dolly, and that was fascinating, and just, even before Shinya Yamanaka had found iPS cells, just nuclear reprogramming is fascinating. Just that you can wipe the epigenome and a cell reverts back to a pluripotent cell. It's natural. It happens naturally, of course, in primordial germ cells. And I spent a bit of time in Cambridge with a really good scientist, Azim Sarani, to learn how reprogramming happens naturally before going to the States then. So that was my academic nuclear reprogramming kind of part of my career. I tend to gravitate towards where I think I'll do the most good, or where I'll have the most value, overall. So, when I was in Boston, I read this book, and the book was How Economics Shapes Science, because I think, as every academic out there will feel, it's a rate limiting step to get the money in. You could do a lot more research if you had the money. So I read this book, and it really spoke to me that, in order to understand how to get science over the line, you kind of have to have the basics of economics in your head. So, I went away and did an MBA and learned a lot of stuff relating to, managing technology and, international collaborations and things of that nature. So after that MBA, then GATE came as an opportunity. And, it had a really stellar board of, that I can engage with and learn from. And it was a really ambitious mission, but it was a really important mission, because you could see how, if the same mistakes were made over and over again, ultimately, things wouldn't be getting over the line, and society wouldn't keep supporting a really important area of research. So, society doesn't know necessarily how much academics suffer for their profession. All they're interested in is, okay, I'll give you so much money. Now you'll give me, a therapeutic or a drug at the end of the day, So we're all linked together in a series of silos, and each silo has, its own value set. So, like in academia, ideation and publications. Then when you move into the manufacturing, it's all about what are the sources of variability, and how do you get something out the end of a process run that is to a consistent standard, consistent specification. So in each of these silos, there's very different foci. There's very different ways of assessing success. And sometimes that leads to cultural friction between the silos as well. So it's, I think sometimes when you have like, an early stage researcher is talking to a regulator or, a manufacturer, they'll look at the regulator and say, you're a nitpicker, you're worrying about all stuff that doesn't really matter. But the regulator has a very broad area of expertise about where things can go wrong when making new medicines, and actually that's really important if you want to make a new medicine. Or if you're talking to a manufacturer and they're talking about, well, we need to upscale this, and the number of cells we grow ultimately determines how many patients we can treat. So like everything is interconnected, and I think I felt that for me, being a jack of all trades and being part of an organization like GATE, which is building a more holistic vision of translation, is actually really important. Because if we stay in our own individual silos, these are just, these are artifactual silos. And they're, if we stay in a silo, sometimes we can be in an echo chamber. And there can be assumptions that aren't being tested in our echo chamber. But we accept as true because we read them over and over again. So, I think one of the things that I've come across in GATE, I've engaged with a lot of clinical immunologists, for example, and, the immune system is a lot harder to control than one would be led to believe if you just stuck to the stem cell literature. There's like clinicians and researchers who are still characterizing aspects of the immune response, and there's so much compensation in the immune response. It's not as straightforward as just ablating a few genes and Bob's your uncle, because even if you do render a cell invisible to the immune system, then when you put it into the patient, the immune system is normally there to oncovigilance, so it's there to remove the cells that are behaving aberrantly and could be a real risk. So if you remove the capacity of the cell to be sensed by the immune system, and that cell starts to go awry, there are aspects of that too. So really, there's a lot of areas of expertise required to make cell therapeutics, and we need to leverage them all in order to get things over the line. And it's important that we mightn't be specialists in all these areas, but it's good to just have an awareness of the bottlenecks, and that's where I think the GATE website is quite useful. And as I say, anyone here listening to this can become a GATE member just by going to gate.global, GLOBAL, and signing up with your email.
Yeah, guys, check out the website. You will learn something, even if you don't want to become a member. I don't know why you wouldn't, but check it out. And really, it hits home what you say, because, there's a lot of scientific journal articles that are published. And the reason why they're published is, they have some clinical ramifications. Oftentimes, it could be the big cure, right? But how many of those things actually really make it to market? I would say 1%, maybe. That's optimistic, because the reality is it's not just the ideation and the hard work, right? But it takes a whole, group of people outside of the science. And you're talking about the regulatory bodies there et al. To really bring it to practice. So it's really nice to have that perspective on the show. And I think all the scientists listening, all the people listening will appreciate what it's going to take and all the hard work that you guys are doing to bring this about. Just on the side here, we have one peripheral question here to ask, because, it seems like you've got a bird's eye view, right? You've seen many aspects of the work, and I think also you have a one perspective of the interface that maybe the scientists on the inside don't have, which is the outside view of the science. What's the biggest misconception about science and scientists that you would like to resolve if you had the chance?
I think you can be a specialist in your area, like in your silo, but going outside and extrapolating the knowledge you have into other areas doesn't always work. So, I think it's really important to know your area of expertise and try and stay within that. And if you don't know, say so. I think I think especially in today's world with social media, I think it's very hard. Like, an expert is only an expert in a finite thing. And like if I start lecturing on, I don't know, like French philosophy, obviously it won't be very valuable because I don't know much. But I think that can be kind of a little bit more nuanced in cell therapeutics. I think one of the biggest things I've learned in GATE is some of the downstream requirements for making a therapeutic, and they're not insignificant. And it's important to have a working awareness of them, because you can literally spend decades refining, making a product, only to realize that it doesn't tick all the boxes and you have to start all over again. So, I think it's important to try and engage in, these organizations that span several different silos. I think, various organizations like the International Stem Cell Banking Initiative, the International Society for Cell and Gene Therapy, for example, ourselves. Like, GATE is like a frigate. It's a small, adaptable kind of organization. We're not an aircraft carrier that ISSCR or ISCTR. But what we can do is kind of at least highlight some of the key challenges that are ahead, and then allow the larger organizations to come in behind and really put more effort and more pressure in that area. But I think one of the good things about being a relatively small organization is that our overheads are low and members can engage on an individual basis and see kind of the workshop material we've produced. And this can help whether you're writing a grant and trying to show that you've thought about the downstream consequences of your work, or if like that you are thinking across silos, and that investors and funding agencies are becoming more sophisticated in asking applicants to be aware of other requirements that are downstream than the early research per se.
Right, guys. Step outside your silos, see the big picture. It's not just about making the experiment work, right? It's about getting it into people. That's what I think every scientist dreams of, one day having their work have a benefit to humankind, whatever that benefit is. And it takes stepping outside of your limited worldview and asking for help sometimes. Thanks for that message, Dr. Sullivan. This has been a really illuminating and fun chat, and we'll have to get you back on the show when you're done with that survey. You can tell us about those 18 respondents and whether or not there was concordance there or not. I have a feeling that we're going to be maybe a little disappointed with the concordance, but maybe some light ahead. All right, partner?
Excellent. Thank you.
Take care, man.
Okay, take care.
All right, people, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on Twitter at STEMCELL or by email at info@stemcellpodcast.com with feedback or to suggest guests. Dr. Sullivan and his group there at the gate trying to get us all on the same page so we can move forward with these iPS cell therapies. Won't be long now. The future is very bright. Tune in a couple of weeks. We'll have another episode talking about the latest developments. Thanks for listening.